Proteins are modified by covalent modification, post-translational modifications that can be detected by mass spectrometry. These data can be used to filter protein interaction networks and also to examine cell signaling pathways in lung cancer. I'd like to acknowledge my colleagues. What we've done is used proteomic data on post-translational modifications, PTMs for short, cluster that data using a machine learning algorithm, TSNE, use those clusters in combination with protein-protein interaction networks and pathways from BioPlanet. The goal is to understand cell signaling pathways in cancer with an eye towards using this knowledge for therapy. Proteins modified by phosphotyrosine, ubiquitination, and acetylation were detected by sequential immunoprecipitation with modification-specific antibodies. 10 cell lines were treated with 4 tyrosine kinase inhibitors as well as a proteasome inhibitor and analyzed to produce these data. The drugs, the tyrosine kinase inhibitors, affected multiple phosphorylations, acetylations, and ubiquitination sites in significant ways. Since signal transduction involves covalent modification of proteins, we hypothesized that patterns in these PTMs, when combined with protein-protein interaction, will elucidate functional signaling pathways in the cells that we are studying. The underpinning nuts and bolts have all been published, and I don't have time to go into the details of these, but they have been extensively evaluated and validated. The result is what we call a co-cluster correlation network of PTMs that includes correlation relationships that are both positive in yellow and negative in blue. Protein-protein interaction networks can be thought of as a hairball. We simplify the hairball by retaining protein interaction edges only when their PTMs co-cluster. Thus, we have what we call a cluster-filtered network, which is useful for examining pathways. We graph these things in Cytoscape. And here's an example. ALK and MET are inhibited by crisatinib, and these other proteins linked on the right are proteins whose PTM significantly changed in response to crisatinib. We're interested in enzymes because they're druggable. This network shows an interesting relationship between glycolytic enzymes and cell signaling pathways that may indicate points of attack. And here is a zoom in showing the individual PTM relationships between EGFR, ALDO-A, and a phosphatase PTPRA, for example. We've looked at a number of shortest paths, including from individual experiments where things are changing in different experiments. And the relationship among the shortest paths is satisfyingly similar to the relationship of the cells used in each experiment and the drugs used to treat them. Okay, to change gears a little bit, now I'm going to talk about BioPlanet. NCAT's BioPlanet is a well-curated amalgamation of what used to be wiki pathways and KEGG and those kinds of things. We're going to use clusters derived from our PTM data to identify pathway-pathway relationships. If the proteins that cluster together contain genes from different pathways, that suggests a pathway-pathway relationship. In this way, we define what we call a pathway crosstalk network. We also have background information. For example, pathway B and pathway C both have a protein in common, and that's a sort of pathway-pathway relationship just defined as a common gene, but we're actually finding relationships between pathways that don't have any genes in common, and we've carefully weighted these different factors. So here's an example of a pathway crosstalk network from the data. If we zoom in on this, we can see a number of interactions among pathways. Let's zoom in further to focus on two pathways that don't have any genes in common, EGFR signaling pathway and transmembrane transporters. Transporters are potentially druggable, so this is worth looking at further. Here's our combined CFN and CCCN, showing the two pathway members identified by color-coded asterisks along with their post-translational modifications painted according to their response to crizotinib. And we can see interesting relationships between solute carriers, neutral amino acid transporters, sodium-potassium ATPase, phospholipid scramblase, and a nuclear poron, along with EGF and RAC, protein kinase A, protein kinase D. So this suggests possible mechanistic insight. If we look at what the response to the proteasome inhibitor, we see different nodes coming up, and different drugs will affect different nodes. So, in conclusion, we've used PTM data from mass spectrometry, and we've constructed networks we call co-cluster and cluster filtered networks to identify data-driven pathway relationships. These data-driven networks are being interrogated further to elucidate more relationships among cell signaling pathways to discover new drug targets for combination cancer therapy. Thank you for your